Looks like we got a fair number of people here who did a DNA test. How many people felt like they got useful information out of it when they did the test? Okay. All right. How many people feel like you really understand how to use the DNA, or like what is legitimate, what's not legitimate? Yeah. So there's a lot of confusion here about how to use your DNA to improve your health. And that's kind of, I'm not going to go too deep into this specific variant or this gene and what worked for me, because there's 20,000 genes and, 100 million SNPs and many other types of variants. The whole idea of precision health is not look at the variants that worked for me or the genes that worked for me, because it's not going to be relevant or too relevant for the ones that work for you. I can tell at the end just what I learned, some of the big things, but essentially this is more of big picture, what to look for, how to understand what's legitimate, what you can do with DNA, what you can't do with it. And so that's the big picture. Just a broad background. I mean, I think everybody here understands that the DNA is the building blocks of what makes you, right? And it's the fundamental information. It's the most important information that you have in you, right? And it's one of the only things that if you get it, the value goes up every year, because the data doesn't change. You buy a car, you buy a phone, you buy anything, the value goes down over time. DNA is just, in my opinion, it's the fundamental thing of what makes you, and the value goes up every year because there's new studies, there's new research, new techniques, new technologies, and I think DNA is extremely important, and precision health in general. The MC stole my thunder over here a bit. But, I had a whole bunch of health issues growing up, and you name it, it was just tons of health issues, and they really prevented me from living a normal life. This was until the age of 26. I had numerous, if we want to go through them, brain fog, fatigue, weakness, lack of motivation, gut problems, insomnia, mental health issues, ADHD, anxiety, depression, joint and skin problems. And so by the age of 26, I was a mess. I felt like my future was bleak, and I actually dropped out of school. I was antisocial. I had no relationships. I was broke. I was unemployed and unemployable. And so what I tried first, I started my life trying out self-help, everything I could get my hands on. I was like, okay, I'm very open to new experiences. I always love to try new things. Let me try self-help. And, you name it. I was doing gratitude journals, just like meditation. And, well, meditation, I wouldn't call that self-help necessarily, but I was doing a lot of, positive thinking, different hacks about how to be more productive or be more calm or this, that, and the other. None of it worked for me. Now, I can't say it doesn't work at all, but it didn't work for me. Certain things may have worked in the short term, but long term, I'm 26. I was learning self-help for years, and I was in a really bad place. So that wasn't my solution. I went to conventional and alternative doctors. They didn't either work for me, and they can work for a lot of people. So that's just a personal thing. They just didn't work for me. And then I decided I'm going to follow other people and see what worked for them, and I'm going to try to copy it to me. And so I tried that with a few different people, because they were saying, like, this works for everybody. So you got to, like, this is the secret solution. You got to do it. You're going to 10x everything in your life, okay? I tried it. Didn't work for me. Through, a couple of them, I had a big health crash. And then I decided, you know what? I'm a gung-ho guy. Like, when I do something, I do it fully. Like, I'm going to do everything but the kitchen sink. And so I read everything that I could, and I tried to do it all together. Every supplement that I read about, every health hack, and you name it. And that didn't either work for a bunch of different reasons. some of them were good for me, some of them were not good for me, and I didn't know what was working, what wasn't working, so that wasn't the solution for me. I like to learn from my failures, and so I learned from self-help that for me, changing my biology was the only route that I was going to change my life. It was by far the most powerful, right? Doctors taught me that I'm the only one that can be in charge of my own health. Nobody else is going to be responsible for my health but me. And the health gurus taught me that we're unique, and what works for one person doesn't work for another, right? I believe these health gurus that what worked for them really did work for them. But it didn't work for me, and I know a lot of people that it didn't work for, right? So as time went on, I met tons of people. I was doing a lot of coaching. It was just very clear that everybody is responding to different things. I respond to different things, and it's clear that two people will be like, this works for everybody, and they have opposite ideologies. So, and from the everything approach, I learned that you don't know what's working for you or against you. You have to try one thing at a time. So these were some of the big lessons that I learned. It took me a while. Again, I was 26, and I started to learn, by the age of 26, I started to learn all these lessons. So the approach that I decided to embark on was, self-learning and self-experimentation. Self-learning involved, I needed to understand how my body, like everything about the body in general, but how my body in particularly worked. And it's kind of where you're starting to see the seeds of genetics, because that is the manual, the blueprint of how your body works. And I also realized that you need to combine this with self-experimentation, right? You can't just blindly follow something. You need you get an idea of a hypothesis, you try it out. You do it one variable at a time, so not everything but the kitchen sink. And it was after I did this approach that I had very big breakthroughs in my health. So I read a lot about health, I developed hypotheses, I did experiments, and I made some very big breakthroughs, and that's when I actually had enough energy to start the website Self-Hacked. But I bought the domain 2 years earlier because I already started, around 2 years earlier, I already started to have the concept of, I needed to fix my own health, but it wasn't at a stage where I had the energy to do that, to actually start something. And so I actually started it 2 years later. And so, the need for precision health, when I started Self-Hacked, my health improved a lot, very big changes, but I wasn't optimal. There were still a lot of things that were unclear, a lot of things that I wanted to optimize. And self-experimentation had its own problems, I realized. Number one is it's very time-consuming because you can only do one thing at a time, and you have to take a megadose of each one to really experience the effect of it. So you really can't do more than like one experiment a day. And if you really think about it, there's just infinite number of experiments you could do, right? Maybe something works for you in the morning, but not at night, and the afternoon. Like this diet combined with this supplement, it was just like I wasn't going to test everything. And so there needed to be a way to get better information, to, a compass in which to better understand what I needed to do next. So I needed a more precise approach, and also many of the experiments did go wrong, had some side effects sometimes when you're taking large doses of things, but yeah. Okay, moving on. So at that point, you can make a lot of improvements with just doing experimentation and learning, and I think that is a great approach for everybody. But luckily, we're in the dawn of a new era of big data, AI, machine learning, genomics, all the omics data, and advances in software that can give you a Rosetta Stone on how to analyze your blueprint and other precision health data, including blood work, your lifestyle. And so software can make that scalable and more easy, more accessible for people who are not spending all day doing experiments on themselves like I was. And so that's kind of what led me to DNA. I realized there needs to be software, not only for me, but for other people as well. And my vision was DNA, lab tests, and environment, because those are the lab tests are mainly blood tests, biomarkers. Those are the three main things that I realized are the critical pieces of information. DNA was the biggest one. There's upsides and downsides to each one of these. DNA, there's huge amounts of information. It can tell you so many things, but you also there's more noise. There's a lot of noise there. Lab tests are very good for tracking over time, but they're not telling you exactly how your biochemistry works. And environmental data can also give you a lot of clues. Of course. So this is when SelfDecode was born. Searchable database for SNPs. I wanted to make sure it was scientifically rigorous, not hypothesis-driven, because what was out there until now was just a bunch of just hypothesis tools, reports. Everything was referenced and accurate, good UI UX, personalized recommendations, real personalized recommendations, and affordable, and also for individuals and health practitioners. This was my goal. That was the reason why I created SelfDecode. Now, I made some mistakes in the field, and I think that that's one of the things I want to impress upon you, what I learned as the CEO of a biotech, precision health, in genetics. What I learned, what were some of the biggest mistakes I made? It turned out that genetics was way more complex than I anticipated. In the beginning, I had hired PhDs in genetics from top universities. I tested their intelligence in some previous, an earlier version of my testing software. And it turned out that just having a PhD in genetics was not nearly enough to actually do genomics, this kind of big data genomics. So that was a mistake that I made. Hiring was really critical. You needed geniuses for this type of stuff. Then there was also studies in 2018 coming out how millions of variants actually influence a trait. Whereas until that point, most of the companies were looking at single variants to try to understand things. And we were guilty of it in the beginning as well. We looked at 50 variants, 100, but it still wasn't looking at millions of variants or many hundreds of thousands. And the new models were showing that this was 10 times more predictive than looking at just extremely significant SNPs or something like that. And so also you needed to use AI and machine learning because you're looking at so much data, and because this is about prediction. I also learned that you needed accurate ancestry, in order to make predictions in genetics. It just turns out that if you don't look at ancestry, then it's pretty much irrelevant, because most of the studies that you see that are based on specific ancestries are only relevant for those ancestries. And it needed to be scalable and integrated, which the version 1 wasn't. So these are because people want, first of all, scalable in the sense that we're looking at so much data. The platform needed to be very dynamic and also integrated with other kinds of data like lab tests. So people look at SNPs. How many people here read a study on a certain SNP and they saw that the risk of something was higher? You read a study, the odds ratio, it's usually it's higher for something. So these studies are almost illegitimate, right? It's something I learned as well. It's because the impact of each individual SNP is very small. And what these studies are looking for, there's a lot of biases built into these studies, but mainly they're doing some data mining because they're looking at the genome, and they're finding some variants that they happen. It's data mining. But besides the data mining, these variants, they're picking up a lot of other things, right? So they're picking up a certain. Usually the study's done certain ethnicity. It's with a group of people that are similar in some way. It's from a certain town in some random place with certain group of people, and also people inherit variants together. So you're also looking at what are all the other variants that are associated with this variant. And so what ends up happening is these risks are very overinflated, and what you end up getting is a lot of contradictions of, like, one study says that this is associated, another study says it's not associated, and anyone who looks at a lot of SNPs and genetics papers will see that the number of contradictions, which make it very complex, but which is why individual SNPs are not very predictive. Which gets you to polygenic risk scoring. So the way that genetics needs to be carried out is what's called polygenic risk scoring. And what that does is it teases out actually the causal variance. So in order to do a prediction, you actually need to understand, you have to, like, if you're taking certain variants, you have to exclude other variants that are just connected. It's called linkage disequilibrium. There are these variants that are just connected to the main variant that you're looking at because they were inherited together. So in order to do the polygenic risk scores, you need to figure out cause from effect, for the cause just from an association. You also need to look at thousands or millions of variants because each variant is going to have a small impact. And so it takes care of the double counting issue, because if you have two variants that both have 50% increased risk, but they're actually just 100% correlated with each other, there's no increased risk, right? And if it's 99% correlated, then you have to take out that double counting. And then you also need a very large data set to validate those models to make sure that the polygenic risk scores are accurate for each model that you're testing, for each condition. Other methods are just not legitimate, meaning like the difference of the impact between these approaches are at least 10x. So the importance of imputation. This is something that most people have not heard about, but it's really important to understand. How many people got a genetic test and had SNPs that were missing from your file that you tried to search up and it just wasn't there? Okay. So anyone who does, like, when I got a test, I tried to look up a lot of SNPs. Like, there were some SNPs for COVID missing from the file. Every day, there's SNPs that were missing from the file. I would read a study, and the SNPs weren't there. It turns out, so you get 700,000 variants usually. That's like the Illumina GSA array. But there's 83 million SNPs, right? And so you're only getting 700,000 variants. But it turns out the variants are inherited together often. And the way these chips are built is that you can predict all of the variants from that set, pretty much all the variants, except the really rare ones, the 1 in 10,000. Those are harder to predict, but you can predict them with 99.7% accuracy using machine learning and AI. There's some, imputation is something that is already on the market. Like, it's always been on the market to some degree or another, but it's usually 95% accurate with looking at, 6 million variants. Our imputation is looking at 83 million variants with 99.7% accuracy. And what that does is it takes care of the problem of missing SNPs. And we have the database of 80 million SNPs as well. So pretty much you could go to any SNP now and look up what your genotype is. Doesn't matter what it is, whether it's in your file or not. With this imputation, everything is going to be in your file. The importance of ancestry. So it does have a very big impact on risk prediction. Variants are inherited together, and good health predictions require accurate ancestry. You do not want to underestimate that. Unfortunately, companies out there, ancestry is actually, so we use ancestry on the back end. It's something we're going to be doing on the front end, but it's not a simple thing to get accurate ancestry. So if you tried ancestry by not the big companies, that's a very easy way to show you that that these companies are not using the correct approaches. So that's something that's very easy to tell. It's like, well, I know, like, I've put my data into various, every company that I could get my hands on that had ancestry. The ones that were, the ones that I saw didn't have a legitimate science team working for them. It just, it said I'm 6% Saudi Arabian, 5% Liberian. I'm 100% Ashkenazic, Jewish, and so it's these were way off. It was just all over the 8% Native American. I'm where is this coming from? Okay, so and by the way, for all 23andMe's faults, they actually did predict that I'm 100% Ashkenazic. So how should genetics be used? Genetics can be very useful at predicting the diseases with the right approaches, of course, and also preventing health issues. So if you know what you're more at risk for, you can take sooner action, but also genetics can sometimes give you a good prioritized list of recommendations. It can help the prioritization of things. This is a really important thing to know. You should not be doing any recommendation just based on a variant. The way that we do the recommendations is, and this is the proper way, is we go through all the research and we sort through the recommendations based on impact and evidence. So what's the impact of this recommendation? What's the evidence for this particular topic? And then those recommendations are prioritized based on genetics, but also based on impact and evidence. So we're looking at many factors. We're not saying you have this variant, therefore you need to do this. That approach does not work. So sometimes I do look at individual SNPs, and this is a personal thing that I like doing. And so when is it a good time to actually look at individual variants? Number one is if all the associations match with you. So if you have a specific genotype, and all the associations just happen to match with you, then it could be something is there. And then I also look at what gene is this involved in? What pathway? Is this something that's relevant to, could it be, does it make sense that this is relevant to the issues that I have? And also how common is the variant? If it's super common, then it doesn't make me different from other people. It's just, everybody's pretty much has it. So those are kind of some of the shortcut solutions that I look at when I develop some hypotheses for myself. So, looking at the team is really important. When I try to hire the team, I looked at other companies to try to see who they hired. It turns out that these companies didn't have anybody, so I couldn't poach from them, which was a very big awakening because I looked at every one of these companies. You could look on the About page or LinkedIn. It's quite easy. I was like, oh, let me see who they have. Oh, it turns out they don't have anybody. Okay, how do they make these claims? I don't know, but it's interesting. But in the end, we were able to successfully hire a very qualified team of people who had the skills to implement these things. And the results showed for it. We tested our polygenic risk scoring models we built models based on the research, and we looked at all the models, and ours outperformed all the industry models. Same with the imputation. So the imputation that we have in the polygenic predictions are best in class. And we're actually licensing that to other companies. I personally think that the space is, my mission is to help as many people as possible, and I think that the way to do that is to make the whole industry conform to a certain, as much as I can, conform to a certain standard rather than, just a lot of BS. Because there's a proliferation of BS in the DNA market and the precision health market, and it's very hard for consumers to understand the difference between what's legit, what's not. And again, even as a CEO of this company, I made a lot of big mistakes myself. And I consider myself a smart guy. So, and well-educated on the topic, so it's very easy to make mistakes here, and how is a regular person going to understand, such a complex topic? But my goal is really to change the industry. So every report has prioritized recommendations with impact and evidence, 150 in-depth reports, lab tracker, and analyzer. I always wanted to make sure that that was part of it. Lifestyle assessments, database of 83 million SNPs to check your variance, and also the top SNPs that are actually contributing to your risk. So these are some of the, these are some of the results of the things that we've done. One second. No. I'm going to go through other things. And also we have a personalized formula as well. Once, based on all, like you can add things from all your reports, your labs, your genetic reports, your lifestyle assessments. You can add recommendations to your regimen, and then you could create a personalized supplement based on what you add. So it's not automatically created for you necessarily. It's just you pick what you want, and then personalized recommendations formula is created. And also, we're going to be doing accurate ancestry as well, which is something only the big companies do. Now, people like to ask, what were some of my personal discoveries? So I'm just going to give some examples because I know people like it. One of the cause of my mental health issues, besides the inflammation, so that's the made worse by inflammation because inflammation activates the HPA axis. There were three things, and my variants did show this. There was low serotonin, so I had a lot of genes. You could see all these genes are related to serotonin in some way or another. And so there are a lot of genes related to serotonin, but all these genes were actually, I had variants that were increasing the risk of mental health issues of some kind or another. And they made sense for me too, because what I did is I tried things that increase serotonin, and that is the most effective thing for me to increase my to improve my mental health, right? And there's a big debate of like, is serotonin really involved in depression or anxiety or this, that, and the other? For me, it was that was the key, right? So regardless of what anyone else thinks, I know for me, it was super important. BDNF was another factor. I did have variants there. So getting sun, getting light, and anything that increases BDNF improves my mood. Anything increases serotonin improves my mood. And then things that modulate the HPA axis also help. And so these were the main pathways that I was able to really do a massive change in my mood. And obviously getting rid of the core health issues, the core inflammation, took me a long way. But even after that, I suffered from mental health issues. It was like periodic because I didn't always know what was suited for me. So I would always take a lot of supplements, and a lot of the supplements did have things that increase serotonin. So it just turned out that a lot of times I was taking these things, but then sometimes I didn't. And so when you don't know what's working for you directly, you're just going to do things in waves. You do it for a couple months, then you stop, right? Now I know this 100% is super critical for me, and I know the class of supplements to take, and so I never stopped taking it after I got that clarity. That's why clarity is extremely important. So supplements that lower inflammation, calm the HPA axis, improve BDNF, and lower serotonin are super, super useful for me. And these are the HPA axis genes. This is the BDNF gene. Again, it's a little technical here, but it is DNA, right? What are you going to do? Dioxins are kryptonite for me. This is one of the things I learned, actually. So, when I improved my health a lot, I went on this binge. I go on binges sometimes, so like different things. Don't ask. But I was going on a binge of sushi, right? 2 weeks of like just sushi every day, tons of sushi. It was like all you could eat sushi, so I took as much as I can and more. And then like I started to get like bad brain fog. And it was a different kind of brain fog than the brain fog that I was suffering years earlier. And I was like, what the hell is going on? There's some toxins in this fish. And I thought it was like heavy metals. So I took things that chelated heavy metals. Didn't help at all, right? So it turns out it wasn't heavy metals. I looked at my genes and I started to look at some genes related to toxins. UGTA, PON1, NRF2. And it turns out that I had variants that were not allowing me to, detox, especially plastics, dioxins. And so I took activated charcoal and it made a huge difference. So ever since then, every time I eat fish, I take activated charcoal, not heavy metal blockers, but just activated charcoal, and no symptoms whatsoever. And it also, when you go crazy with one thing, like, so like just eating fish for 2 weeks, it actually gives you a strong understanding of like, okay, this is impacting me, right? And then ever since then, if I eat fish once, I'll start to notice it. It's much lighter, of course, but I do notice it, and then I take the activated charcoal. And microchitosan also works, by the way. Another thing that I learned was the source of my food sensitivities. So it was related to the cannabinoid system, and also the Th1 and Th17. So essentially, my immune system was just overactive because of these systems, cannabinoids, THC. They suppress the immune system. If you smoke marijuana a lot, you'll probably get sick. It's because they suppress the immune system. They also suppress the Th1, Th17. It's actually really good for my food sensitivities, but not so good for getting work done. But there's actually other ways to improve the cannabinoid system that don't involve getting high. These are some genes: STAT3, IL-17, CNR1. And then, the source of high cholesterol, it turns out that if lowering saturated fat, that's what I got from my genes, also from experimentation. And fiber also helps a lot. Now, and by the way, just something cool, a random tidbit, and we could go through some questions that people have. But there's something called Mendelian randomization that helps you. It's actually a technique part of genetics that helps you tease apart cause and effect. So we do a lot of lab stuff, and like we have a lab platform, and when you're doing lab stuff, it becomes very complex. There's a lot of contradictions, and what's causal, what's not. It turns out, so these Mendelian randomization studies look at people who have genetic variants that increase their LDL cholesterol, vitamin A, any kind of nutrient, any kind of lab marker. And that is an independent variable because it's just the genes that are doing it, right? It's variants that are independently increasing this marker. Then by testing those people, you can see, and especially if it corresponds to other information that you have, you could see if it's actually a cause. Turns out that LDL cholesterol is a cause, but that's is causal for heart disease. You learn a lot of interesting information about supplements. It turns out that a lot of supplements are harmful in certain ways as a cause. And again, it's hard to get that from observational studies because the observational studies are people who have high selenium, for example, are eating a lot of nuts and vegetables. So it's not going to come up that they have a higher risk of diabetes, even though that actually increases the risk of diabetes, people who have naturally higher selenium regardless of their diet. That's a random tidbit, though. So I was actually thinking about, should I go through some of these Mendelian randomization, but I said, nah. Okay, does anyone have questions? Sure.
You said you require really large data sets to validate your data when using AI. So what kind of data sets are you using and where do you get that data from?
The UK Biobank and also our internal data sets. But the UK Biobank is 500,000 data sets with traits and conditions, so you could see the genetics and the traits, and so we can validate the models using those. Yeah.
You said don't look at individual SNPs because they don't really give you the true picture, but then there are SNPs like the double 4 allele with the APOE4, which is kind of highly associated with Alzheimer's. So how do you feel?
Very good question. Those are different, right? Those are like increasing your risk by like 27 times or 13, 15 times, something like that. That is in a different category, right? So when I say don't look at individual SNPs, there are certain SNPs that they've been validated across ancestries by multiple studies. They're going to have some impact, but you still need to look at them in relation to a lot of other SNPs. You can't look at them in isolation. ApoE, the ones that go into the medical establishment generally they're so powerful, so it's just obvious that this alone, BRCA would be a good example of that. Um, MTHFR is iffy. Yeah.
Thanks a lot, Joe. I was wondering, whole genome sequencing is becoming increasingly cheaper and cheaper. Do you think at some point this will be better to replace SNPs into whole genome sequences if you can? Or do you think this is going to be the gold standard for a while?
So that's a great question. Whole genome sequencing is going to eventually replace SNPs. The thing is that at this stage, companies are not really analyzing the rest of the stuff besides SNPs, generally speaking, in whole genome. So it's not that relevant. It's going to be more relevant over time, maybe the 3-4 year range. And so with imputation, it kind of extends the lifestyle of these SNP chips, these genotypes. And it's way cheaper. And usually when you're getting the whole genome, there's companies that sell it for cheap, but they sequence it in China. So the Chinese government owns your data, but not a problem. A minor issue.
Hey Joe, yeah, a little bit related to that last point you made. So you made a mention of 23andMe earlier and it having a few shortcomings. I know in the past there was a bit of controversy that 23andMe might be using genetic data for other purposes. It's got a lot of people scared. Just curious, in your case, do you use your customers' genetic data outside of analysis?
No, absolutely not. So we just use it for our analysis, but we do not sell the data. 23andMe's business model was built on that premise of selling the data to pharmaceutical companies, and they've done it with Glaxo. That is not our model, right? So we're very strict about privacy and security and selling data as well, and not making sure that the data is private and not selling it. So yeah, it's a very different model. The truth is that generally smaller companies are not going to be like, just because 23andMe has like 13 million samples, and so the game keeps on, like, if you want to sell them, it's just like they'll buy from 23andMe. We have 100,000 samples. It's like, I mean, we don't, but that's not our business plan. That's not our customer base. We know everybody doesn't want it. We don't believe in that. We're very against it. So, yeah.
Your last comment about selenium, there, you say that it contributes towards the diabetes epidemic, but German school of thought, they say they are prescribing selenium in case of autoimmune diseases like Hashimoto's.
Yeah. So, I was going to put a lot of stuff in here, but I knew it was going to be too long. You really have to look at this Mendelian randomization stuff. It's a game changer. Things like for vitamin D, something that's the only thing it turns out that the Mendelian randomization validated was multiple sclerosis that it helps with, and like maybe Alzheimer's and respiratory infections. Everything else was just, people who got sun were healthier. They got outside more, probably, right? So it's a long conversation, but this is the only game in town, really, with that, because you can't do clinical studies, like really long-term clinical studies on most of these things. Like, you're not going to give selenium for 40 years and see who gets diabetes after 40 years. So the Mendelian randomization is really the way to go. Okay. Looks like we're done. Do we have time for one more? No? One more. Okay.
What supplements do you take to increase your serotonin?
That's a great question. So, I mean, this is not actually my main business is SelfDecode, but I actually just started a supplement just because I took the stuff that worked for me. I'm like, I'm going to make this into a supplement because people are, and the people who I think are low in serotonin, and basically have a profile similar to me, I'll recommend it. But essentially, I put all the main stuff in the supplement. It's rhodiola, 5-HTP, bacopa, and ashwagandha, but you have to get certain extracts of those. So that's another thing, is the extracts vary very, very much depending on, like, different kinds of ashwagandha will do different things. Certain kinds will work on serotonin, certain kinds will not. So I've taken like 15 pills of certain kinds of ashwagandha, the KSM ones, no effect. And then I've taken 3 pills of some other ones. There was a noticeable effect. So it's very clear that just based on my own experiments, that the different extractions are very important. Okay. Thank you very much, everyone.